Okay, okay. Before we do anything else, I need everyone to sit with a number for a second. 50 to 70 billion. That is how many cells in your body are going to die today, not because you're sick, not because you ate something questionable, on purpose, by design. Your body, right now, while you are listening to this, is executing a coordinated, controlled, biochemically precise mass termination event, and you have not been consulted at any point in this process. You had no input, you received no memo. 50 to 70 billion of your own cells have made a decision about their own futures without asking you. And the decision was, today's the day.
That is an accurate framing, yes.
Dr. Seeley, starting with validation. What a day to be alive, which apparently is a privilege my cells are actively renegotiating.
And it's not just death, it's elegant death. That is the part that breaks my brain every time I think about it. It's a program. It runs subroutines. It has checkpoints and fail-safes and redundancy. It's basically the most sophisticated piece of software in the known universe, and the output is just the cell neatly disassembling itself from the inside, packaging its own remains into tidy parcels, and quietly waiting to be collected. No fuss. No alarm. Just stun.
It is mildly poetic that the most sophisticated thing your cells do is choose to stop.
50 to 70 billion units a day. That is a scale operation. I've seen logistics companies with worse throughput than that. If someone could productize controlled cellular elimination, targeted demolition as a service, subscription-based, I'm not even joking, I would fund that in a heartbeat.
My aunt's house was demolished once.
I suspect that is the beginning and end of that observation.
There was a pigeon inside.
Is the pigeon?
It was fine.
Let's establish terminology before anyone further destabilizes this conversation. What Bobby described is called apoptosis, from the Greek apoptosis, meaning falling off, as leaves fall from a tree. It is a specific, genetically encoded program. It is emphatically not the same as necrosis, which is what happens when a cell is killed by external damage. Infection, physical trauma, toxins, prolonged oxygen deprivation. Necrosis is uncontrolled. The plasma membrane ruptures, the cell's contents spill into surrounding tissue, and the immune system treats it as a full emergency. Inflammation, recruitment of immune cells, tissue damage. It is messy, by definition. Apoptosis is the opposite. The cell shrinks. It packages its DNA and organelles into membrane-bound vesicles called apoptotic bodies. Neighboring cells and macrophages phagocytose those bodies before any contents are released. No spill. No inflammation signal. No alarm.
So necrosis is like a car crash on the motorway. Everything stops, there's debris everywhere, emergency services scramble, the whole road is affected for hours.
That is a reasonable analogy.
And apoptosis is like the car just quietly indicates, pulls into a lay-by, turns off the engine, and a tow truck arrives and removes it before anyone even noticed it had stopped.
That is surprisingly good, yes.
The tow truck is a macrophage. The macrophage engulfs the apoptotic bodies via phagocytosis, breaks them down in lysosomes, and recycles the components. It is genuinely one of my favorite processes in all of cell biology. I have watched macrophages engulf things under a fluorescence microscope at 2 a.m. for entirely recreational purposes, and I have no regrets whatsoever about any of it.
Macrophage logistics, cellular recycling at scale. I'm genuinely thinking about the supply chain implications.
Please don't.
Okay, but how does the cell actually decide? What is the mechanism? How does a cell sitting there, doing its job, get to the point where it goes, right, today's the day?
There are 2 main pathways, and they converge on the same executioner mechanism. The intrinsic pathway is triggered from within the cell: irreparable DNA damage, prolonged oxidative stress, loss of survival signals from neighboring cells, developmental instructions. The extrinsic pathway is triggered from outside: death ligands binding to death receptors on the cell surface. The most studied is Fas ligand binding to the Fas receptor, which is how cytotoxic immune cells kill infected targets. Both pathways converge on a family of enzymes called caspases.
Caspases. Please say that word again. Caspases. It sounds like a villain. It sounds like the name of a villain's organization in a film where the villain has very good scientific advisors. And the name is completely warranted.
Caspases are cysteine aspartide proteases, enzymes that cleave other proteins at specific aspartate residue sequences. They exist in the cell in inactive precursor forms called procaspases. When apoptosis is triggered, initiator caspases, caspase 8 via the extrinsic pathway or caspase 9 via the intrinsic, are activated by cleavage. Those initiators then cleave and activate the executioner caspases, primarily caspase 3. And caspase 3 is where the program becomes irreversible.
Irreversible is doing a lot of work in that sentence.
Intentionally. Caspase 3 cleaves the structural proteins of the cell, the cytoskeletal filaments, the nuclear lamins that support the nucleus. It cleaves and inactivates DNA repair enzymes, which removes the cell's last-ditch ability to fix itself. And it activates a DNase called CAD, caspase-activated DNase, which fragments the genomic DNA into pieces of precisely 180 to 200 base pairs. Precisely. Every time. That regular laddering pattern is a forensic signature of apoptosis. Pathologists use it to confirm programmed cell death in tissue samples.
The cell, while dying, produces evidence of its own death in a very specific, recognizable format. That is, the cell is writing its own death certificate.
I would not phrase it that way in a paper, but the structural analogy is not entirely wrong.
There is something about that I find genuinely moving, and I cannot fully explain why. The cell in the process of ending leaves a legible record of the ending, and outside observers can read it. I think about that more than is probably healthy.
The fact that Kelpie finds a mass cellular termination event moving is mildly the least surprising thing said today.
You mentioned the intrinsic pathway, the one triggered from inside the cell. What is actually going on in there? Like, what's making the decision?
The decision is made by the balance between two opposing subfamilies of proteins in the BCL2 family. Anti-apoptotic proteins, BCL2 itself, BCLXL, MCL1, sit on the outer mitochondrial membrane and actively prevent it from being breached. Pro-apoptotic proteins, BAX and BK, want to form pores in that same membrane. A third group, the BH3-only proteins, BIM, PUMA, NOXA, are the sensors. They detect cellular stress, and when activated, they bind and neutralize the anti-apoptotic proteins, tipping the balance towards BAX and BAC.
It's a molecular argument, running constantly, every cell. Anti-apoptotic proteins saying, not yet, the cell is fine, we're holding, and pro-apoptotic proteins accumulating as damage builds up, waiting for the moment the balance flips. You are being kept alive right now by the fact that your BCL2 proteins are currently winning their argument. In every one of your 37 trillion cells, simultaneously.
It is mildly alarming to frame existence as 37 trillion simultaneous ongoing negotiations with an uncertain outcome.
When BAX and BK win, when they form those pores in the outer mitochondrial membrane, the mitochondria releases cytochrome C into the cytoplasm. Cytochrome C is normally a component of the electron transport chain. Out of context, in the cytoplasm, it binds a scaffold protein called APAF1, and together they form a wheel-shaped structure called the apoptosim. The apoptosim recruits and activates caspase 9. Caspase 9 activates caspase 3. The cascade is running. There is no pulling it back.
The mitochondria, the ATP production organelle?
Please don't finish that sentence.
I wasn't going to say the thing.
You were absolutely going to say the thing.
I was going to say the thing. The mitochondria releases a component that kicks off the entire death program of the cell. The mitochondria is literally the cell's kill switch.
Among its functions, yes, which is part of why mitochondrial health is so central to cellular survival.
Mitochondria-focused wellness. I feel like this is already a market. It is already a market, isn't it?
Several billion-dollar markets, yes.
I had an argument once. It lasted 6 years.
Did you win?
I forgot what it was about.
You said cancer cells disable this. How exactly does a cancer cell jam the apoptosis mechanism?
Several ways. Some cancers, particularly follicular lymphoma and CLL, have a chromosomal translocation that places the BCL2 gene next to a highly active promoter. The anti-apoptotic protein, BCL2, is massively overexpressed. The balance is permanently skewed. Pro-apoptotic signals arrive, the cell detects damage, the BH3-only proteins are activated. But BCL2 is present in such abundance that they cannot neutralize it. The pores never form. Cytochrome C stays in the mitochondria. The caspase cascade never starts. The damaged cell continues dividing.
And that's just BCL2. Other cancers silence Bax directly, methylating the promoter so the gene isn't expressed. Some downregulate APAF1, so even if cytochrome C is released, there's nothing for it to bind. Some express decoy death receptors that absorb extrinsic death signals before they can activate caspase 8. Cancer has independently evolved at least half a dozen different mechanisms to block apoptosis, because apoptosis is the single biggest threat to its existence.
Cancer is genuinely terrifyingly clever.
It is mildly more accurate to say evolution is terrifyingly indifferent. Cancer doesn't plan, it just keeps the variants that survive.
Which is worse, honestly.
The therapeutic response to this is Venetoclax. It is classified as a BH3 mimetic. It occupies the same binding groove on BCL2 that the endogenous BH3-only proteins would use. It outcompetes them for the site. With BCL2 blocked, the pro-apoptotic proteins it was sequestering are released. Bax and BK form their pores. Cytochrome C is released. The cell executes apoptosis, the apoptosis it had been biochemically suppressing for potentially years.
So the drug is just removing the cancer's cheat code? Just undoing the specific thing the cancer did to become immortal?
In a targeted mechanism-specific way, yes. Venetoclax is approved for CLL and certain AML subtypes. Response rates in previously untreated CLL in combination with obinutuzumab are in the range of 90% measurable response. That is a drug that works because someone traced the exact molecular mechanism of cancer's survival advantage and designed a molecule to block it at its source.
That is an extraordinary value proposition. You identify the specific protein the cancer needs to survive. You make a molecule that binds it and nothing else, and the cancer kills itself. The mechanism sells itself.
I have been reading the crystal structure of BCL2, bound to Venetoclax, for reasons I will not fully disclose, but which are, I want to stress, oriented towards understanding and not replication in an uncontrolled environment.
The qualifier at the end of that sentence is doing very significant work.
Okay, so cancer cells are immortal because they disabled apoptosis and they're replicating forever. But normal cells, even healthy ones that aren't cancerous, they're not immortal either. They have a limit, right? A built-in expiry?
Yes, every chromosome ends in a repetitive DNA sequence, T-tag, repeated hundreds to several thousand times, called a telomere. Its function is structural and protective. Telomeres prevent the chromosome ends from being recognized as double-strand breaks, which would otherwise trigger the DNA damage response and either halt the cell cycle or initiate apoptosis.
They're protective caps, like the aglets on the ends of shoelaces, the little plastic tips that stop the lace fraying. The telomere stops the chromosome from fraying. Except the telomere itself gets shorter every time the cell divides, because of a fundamental limitation of DNA replication.
The N replication problem. DNA polymerase requires an RNA primer to initiate synthesis, and it adds nucleotides only in the 5' to 3' direction. At the very tip of a linear chromosome, the final RNA primer cannot be replaced with DNA after it is removed. A small stretch of sequence is therefore lost with every replication cycle. Not in the coding regions, that's what the telomere is for, but the telomere itself shortens progressively with every cell division.
So cells have a countdown built into their chromosomes.
Effectively, after approximately 50 to 70 divisions, a number first described by Leonard Hayflick in 1961, now called the Hayflick limit, the telomere becomes critically short. The protective cap fails. The chromosome end is exposed. The cell detects this as DNA damage. It cannot safely divide without risk of chromosomal fusions or deletions. At this point, most cells enter a state called senescence.
Not death, but not quite life either.
The senescent cell does not divide, but it is metabolically active. It continues secreting proteins, consuming energy, responding to signals. It has withdrawn permanently from the cell cycle, but it has not undergone apoptosis.
It is mildly accurate to describe a senescent cell as a retired employee who refuses to leave the office and has started interfering with everyone else's work.
More accurately, senescent cells develop what's called the SASP, the senescence-associated secretory phenotype. They secrete inflammatory cytokines, proteases, and growth factors into the surrounding tissue. In the short term, this can attract immune cells to clear damaged tissue. In the long term, accumulating senescent cells create a chronic low-grade inflammatory environment that is now strongly associated with age-related tissue dysfunction in certain cancers.
There's a whole field now focused on senolytics, drugs that selectively clear senescent cells. The idea is that if you periodically purge the accumulating senescent population, you can extend health span. Mouse studies are genuinely compelling. There are people in extremely expensive biohacking facilities taking Venetoclax as a senolytic off-label, and I have many thoughts about that experiment that I am keeping largely to myself.
I know 3 of those people. One of them texted me about it last week.
My grandfather was very old once.
Is he still?
He's fine. He's gardening.
So cancer cells, if normal cells have a telomere countdown, and senescence is what happens when the countdown ends, cancer cells must be doing something to the countdown.
Approximately 85 to 90% of human cancers reactivate an enzyme called telomerase. Telomerase is a reverse transcriptase. It carries its own RNA template and uses it to add T-tag-G repeats back onto chromosome ends. It refills the telomere. In normal somatic cells, the TERT gene, which encodes the catalytic subunit, is silenced. Germ cells and some stem cells express it. Cancer cells reactivate it. With telomerase running, the telomere does not shorten. The Hayflick limit does not apply. The cell can divide indefinitely.
Combine disabled apoptosis with active telomerase, and you have a cell that cannot be told to stop and cannot be told to die, and has removed the one natural expiry mechanism that would have ended it on its own. It is, by any reasonable definition, biologically immortal, and it is also broken in nearly every regulatory sense that matters to a multicellular organism.
I want to tread very carefully here. But immortal cells that can be cultured indefinitely, at scale, are genuinely extraordinary research tools. There's an obvious commercial interest in cell lines like that. I assume someone has figured this out.
There is a deeply uncomfortable historical dimension to that observation.
Is this the HeLa cell thing?
Yes. In 1951, Henrietta Lacks was admitted to Johns Hopkins Hospital in Baltimore for cervical cancer treatment. A sample was taken from her tumor, without her knowledge and without consent, which was standard practice at the time. Those cells were given to a researcher named George Gay, who had been trying for years to establish a stable human cell line for research. Every prior attempt had failed. These cells survived. They divided. They kept dividing. They became the first immortal human cell line, designated HeLa after the first 2 letters of her name.
Those cells were used in the development of the polio vaccine. They went to space aboard early Soviet satellites to study the effects of microgravity on human cells. They have been used in over 110,000 published research studies. They are growing in laboratory incubators right now, on every inhabited continent, in thousands of separate facilities. Henrietta Lacks died in October 1951. Her cells have been continuously dividing for 75 years.
I do not know whether that is one of the most amazing things I have ever heard, or one of the most ethically alarming.
It is mildly both, and the fact that it took until 2013 for the NIH to reach a formal agreement with the Lacks family about how her genome sequence would be used is relevant context.
There were licensing arrangements that generated significant commercial returns for entities other than the Lacks family. I have a lawyer present, even in thought, and I will say only that the situation has evolved.
What gets me, on the pure biology, is that HeLa cells are now so genetically distinct from the original tumor that they are arguably a separate species. 75 years of accelerated evolution in culture conditions has produced a cell line that differs dramatically from normal human cells. They've been called HeLa hominis by some researchers. Henrietta's cancer cells have speciated.
If you leave something out, it keeps going.
That is remarkably accurate.
That is exactly what happened. The cancer cells were left out of Henrietta Lacks, and they kept going. Ugh.
It is mildly the most precise thing said in this conversation, which is saying something given Dr. Seeley is present.
Okay, I want to take stock of where we are. We have apoptosis, cells dying on purpose, elegantly, 50 to 70 billion of them today alone. We have necrosis, messy death from external damage. We have senescence, cells that hit the Hayflick limit and retire but don't quite leave. And we have cancer cells, cells that have broken all of this. They've disabled apoptosis, they've reactivated telomerase, they've ignored every signal telling them to stop. So the question is, why don't we all just get cancer all the time? Because it sounds like the failure modes are everywhere.
That is the correct question, and the answer requires admitting that what I've described so far, the internal apoptosis machinery, the telomere clock, is not the only system standing between a damaged cell and a tumor. There is an external surveillance system, a patrol, and it is watching for exactly the kind of cellular misbehavior we've been discussing.
Say it.
The immune system,
Of course, it's the immune system.
specifically a process called immune surveillance. Oncologists have understood for some time that protocancerous cells arise in your body regularly, every day, likely thousands of times. Replication errors, oxidative DNA damage, stray mutagenic exposure. The cells that result have abnormal phenotypes, disrupted surface proteins, unusual metabolic signatures, stressed molecular patterns. And natural killer cells, NK cells, are continuously patrolling tissue, looking for exactly those signatures.
What are NK cells actually looking for? How do they recognize that a cell has gone wrong?
Primarily the absence of a signal rather than the presence of one. Every healthy cell in your body displays protein complexes on its surface called MHC class I molecules. These present small peptide fragments from proteins being made inside the cell, a continuous molecular broadcast of what the cell is currently doing. NK cells check that signal. If MHC class I is present and normal, the NK cell moves on. If it is absent or downregulated, which cancer cells frequently do, partly to avoid detection by cytotoxic T cells, the NK cell reads that absence as a threat signal.
And then it does something extraordinary. The NK cell synapses with the target cell, deploys a protein called perforin that polymerizes and punches a channel directly through the target cell membrane and injects a cocktail of serine proteases called granzymes into the cytoplasm. Granzyme B directly cleaves and activates, you already know this, caspase 3. The NK cell makes the cancer cell kill itself using its own apoptosis machinery. The exact same mechanism the cancer cell was trying to suppress.
The NK cell uses the apoptosis pathway to kill the cell that was trying to escape apoptosis.
The symmetry is not lost on me.
It is mildly elegant in a way that makes me deeply suspicious of what the immune system was designed to compensate for.
So enhanced NK cell surveillance would catch more early-stage cancers before they establish. Is that the logic behind cancer immunotherapy? I have been in 3 pitches in the last 18 months where the deck opened with a version of this slide.
That is the foundational logic, yes? But I want to be precise about something critical. The immune surveillance I've described is extremely effective in early stages. Most protocancerous cells, the ones that arise from replication errors and accidental DNA damage, are destroyed before they ever divide more than a handful of times. The immune system is genuinely the primary reason you are not accumulating tumors constantly. But the reason cancer ultimately wins, in cases where it wins, is not that the immune system never noticed. It is that the cancer eventually learns to hide.
Cancer that survives the NK cell patrol has been selected for immune evasion. It re-expresses MHC class Yahoo, but loads it with peptides that look normal. It starts expressing surface proteins called checkpoint ligands, PDL1 for instance. that don't just hide from immune cells, but actively switch them off. PD-L1 binds the PD-1 receptor on T cells and delivers a suppressive signal that tells the T cell to stand down. The cancer is not just hiding, it has learned to turn off the guards.
That is genuinely sinister. The cancer isn't just evading the immune system. It's using immune system machinery to actively suppress the immune response against it.
Which is why checkpoint inhibitors, drugs like pembrolizumab that block PD-1, have transformed certain cancer treatments. You block the suppressive signal, the T cells are released, they can see the tumor again. But the full mechanism, the checkpoint biology, the arms race between immune evasion and immunotherapy, that is a full episode. Because it deserves one.
Pembrolizumab is a $40 billion drug. I want to flag that for context.
I have been trying to understand the structural basis of PD-1 checkpoint suppression for 11 days, and I have a lot of thoughts that I am very ready to share.
I have begun a list of things Kelpie has spent more than a week on in a shed context, and I expect it to become a long list.
Wait, Dr. Seeley, you actually just did a cliffhanger. You literally just went, but that is a story for another day. You did the thing. You did the podcast cliffhanger thing.
I provided appropriate scientific context for an ongoing mechanistic question that merits extended treatment.
That is a cliffhanger.
It is a cliffhanger.
Incredible, okay? Potter, as is the tradition, as is the sacred tradition of this podcast. Closing thoughts?
The cell has a vote it doesn't know it's casting. Every moment, keep going or stop. The vote is counted in the ratio between proteins arguing in a language of molecular shapes. When everything works, the damaged cells lose the vote and disappear quietly. 50 to 70 billion of them every single day, and you never feel it. When cancer works, it rigs the count. It silences the stop signals. It overexpresses the keep-going proteins. It refills its own clock. It becomes the one cell in the committee that refuses to be outvoted. But the body expected this. There's a patrol that doesn't check for what a cell is doing. It checks for what a cell is broadcasting. When the broadcast goes quiet, the patrol notices. Most early cheaters get caught and killed using the very mechanism they were trying to escape. The ones that survive have learned to forge the broadcast. That is the next problem.
I need to lie down.
That was, word for word, a more coherent summary of cancer biology, apoptotic evasion, and immune surveillance than I have encountered in several recent review articles. I would like to understand when exactly you assembled that.
I was listening.
But when? We were all here. You spent most of the time talking about your grandfather.
He's very old. It leaves time for thinking.
It is mildly extraordinary that the person who contributes least to each discussion is invariably the most prepared for it.